Hey guys, it's Miss Pham again. You should have already finished the notes for transcription, the actual process of copying DNA into mRNA, messenger RNA coding. Now this is the next step. So the whole point of this is to make proteins, and the first part is the copying down of the DNA sequence, the instructions. Now we're actually going to make them, or the body can make them. So, again, function, a process to make a protein based on the sequence of mRNA. At the end of transcription, the mRNA molecule is sent out of the nucleus and into the cytoplasm. Again, so that's just a little reminder of the step right before this starts. And the reason we're doing this at ribosomes is because that's where we make proteins. I briefly mentioned this yesterday, but I really wanted to emphasize it today. I told you guys we were going to talk about it tomorrow, and here is tomorrow. Each mRNA sequence is read in triplets. Think of it like a word that a three-letter word. So this triplet that I have here, let me zoom in on it. AUG, so that's one triplet. CCG, 2. AUG again, 3. CGU, 4. UAA, 5. Okay, 5 triplets. Each triplet is called a codon. That's very important that you understand that vocabulary. Commit it to memory. A codon is a triplet sequence in mRNA. Because once we keep going, you're going to hear another vocabulary word. And here it is. Once the mRNA gets to the ribosome, tRNA. And what was tRNA again? Transfer RNA. I'll give you time to think about it for a second. See if you can answer the question. So the mRNA is at the ribosome. Excuse me, guys. Now we have to wait for the tRNA. Alright, so the tRNA has to find its matching triplet. It'll match with a codon. And that codon will have something called an anticodon. So that's the 3 letters that will pair up with the sequence in mRNA. And so what you should have next in your notes is this picture right here. Alright, so it's the same, the mRNA sequence is the same as above. We're going to just stay consistent in that aspect. So the codons are this row up here, making this row at the bottom the anticodon. Alright, so notice, I'm just going to go over the first one. We know that A will pair with U if we're talking about RNA, right? And vice versa, U is going to pair with A. Now, G and C will always pair with each other as well. Okay, so what's going to happen is A, you see in this codon is AUG, A is pairing with U, U is pairing with A, G is pairing with C. You see how that happened? Okay, let me go through it again. This is a codon, AUG, so then its anticodon is going to be UAC, because that's what's going to pair up with that triplet. And then it just keeps going. CCG is going to pair with GGC. We're going to be doing a lot of practice for this particular thing over the next day or two. Okay, so the next step, once the anticodon has attached to the correct codon, right, once AUG has UAC attached to it on the tRNA side, you're going to get an amino acid attached to it on the bottom, just like in this picture. So there's your amino acids, right? And then your amino acids, once several of them get into a chain, they're going to make a protein. Now, when I wrote these notes originally, I must have not been thinking clearly. It's not that it takes 20 amino acids. I don't want you to write that particularly. It doesn't take 20 amino acids. There are a total of 20 amino acids. 20 different types. And I wonder if I can, okay, sweet. There are 20 amino acids, and those code for proteins. So the protein isn't 20 amino acids. It could be 7, it could be 50, it could be 12, it could be any series of numbers that make it a 3D shape. But there are only a total of 20 amino acids. So it could be, 20 amino acids starting from the first one to the last one, or maybe it's only 10 amino acids and some of them are repeating. There's all kinds of different combinations. The possibilities are endless, but there are only 20 amino acids, and I want you to really pay attention to that when I go to the next step. Alright, so just to review, translation, I'm going to scroll back up. Oh, that's stuck there. That's okay, we'll fix it later. Translation is a process of actually making the protein. So if we were equating the, oh, now it's stuck. If we were saying, like, from our, the cookie that we did, the making of the cookie, we said transcription would be the process of one of your lab tables partners, table partner? One of your group members wrote down the recipe, went out into the hall and wrote down the recipe and came back in. That would be transcription. And then you guys, as a group, getting the milk, the sugar, the butter, the oats, the chocolate chips, all that stuff, that's going to be your translation process. And our final product was cookies. And the cell's final product is a protein. And how it does that is it has your mRNA sequence, which is the recipe, then the tRNA has the anticodon, that's the matching. It's kind of, the anticodon is basically the cell's way of making sure that they're following the recipe. So like for you guys, when you went to go get your ingredients, you just checked it off your recipe list. That's kind of like a checkoff. If I have UAG, I bring an amino acid that pairs with it by making sure it's attached to UAC, and that's a check. Then we go to the next one, check, next one, check, next one, check, till we get the protein that we need. Alright, so that's the end of translation. It's really just the coding. We'll talk more about the actual sequence of amino acids and how you can find those out in another video. Thanks guys. Thanks for paying attention.